Welcome. Thanks for inviting me to present. So we're well into the post-prandial insulin surge, I'm sure, and you're all feeling a little sleepy, so I thought I'd do a little get-to-know your audience exercise. Let's start with an easy one. Hands up if you believe that cell and gene therapy has the potential to transform medicine as it's practiced today. Anybody whose hand is not up, why the hell are you here? Okay, let's do another one. Who thinks that they could explain the opportunity for T-cell, engineered T-cells beyond CAR T? Okay, that's a little more difficult one. Okay, I'm not going to ask you to do that. Two of the people who put their hands up work for my company, so that's great. And I would expect they would know that. The third one, who's ever heard of Adaptimmune? Okay, well, that's a little better. So I'll try and pitch this at an appropriate level, and let me know if I'm not doing that. Forward-looking statements available on our website. Adaptimmune, let me ask you one more question. Who was doing cell and gene therapy 20 years ago? Excellent. This is a very long-lived field, and the successes that we are having at the moment as a field are based on an enormous amount of work that goes back 20 years, and that is true for any new modality of therapy. If you take biopharmaceuticals, you take oligonucleotides, you take cell and gene therapy, they all have long histories. Adaptimmune was there pretty much in the early stages. In 1999, a company called Avidex was spun out from the University of Oxford based on the research from our scientific founder, Bent Jacobsen, who discovered how to stabilize T-cell receptors so they can be studied in a solution. Unlike antibodies, T-cell receptors, the native binding domain of a T-cell, do not exist in a structure that is not bound to the cell surface. And because they do not exist in a structure that's not bound to the cell surface, they do not exist in a soluble setting. You cannot do routinely the level of engineering that everybody's been doing with antibodies for a long time, and that's why we don't have a whole field of medicine defined by T-cell receptors. Bent was successful in working out how to solubilize T-cell receptors and keep their conformational stability. And that work led to the foundation of a company called Avidex, which was successful for several years and then got acquired by Medigene. And in 2008, Medigene decided that it no longer wished to participate in this particular field. And Adaptimmune spun out with the cell therapy rights to all of this fundamental intellectual property that goes all the way back into the early 90s and into the late 90s and the early 2000s. Initially, we were essentially a semi-virtual company, working with Carl June at the University of Pennsylvania, working on a program called NYESO, which I'll talk about in a second as well. And then we took advantage of the boom in cell and gene therapy as it became a real thing. We IPO'd in 2015. We did a partnership with GlaxoSmithKline in 2014. And we've raised approximately $500 million prosecuting NYESO and our wholly owned programs. It brings us to 2018, which I think you'll see is going to be a landmark year for Adaptimmune. We're currently just over 400 employees based in the UK and the US. In the UK, we have probably the preeminent protein engineering team focused on T-cell receptors, 140 people on target discovery all the way through to T-cell and candidate selection. In the US, we have manufacturing, clinical, regulatory, and corporate functions split equally. And just up the top in Stevenage, our latest addition at the Cell and Gene Catapult facility in Stevenage, our vector manufacturing facility is standing up as we speak, and we'll be producing vector next year. To explain just very briefly what we do is, unlike a CAR T, where you stick an antibody onto the side of a T cell and target it at a cell surface protein, we engineer the T-cell receptor, the native signaling, the native binding domain and signaling machinery of the T-cell. That has a number of advantages. If you use an antibody, you can only target things that, generally speaking, there's always exceptions, but you can generally speaking only target things that antibodies see. Those are cell surface proteins. There are very few cell surface proteins that are specific to cancer. So the CAR Ts, as we know, target CD19 or BCMA. Those are largely lineage antigens. and they conform to the rule that if you have a malignancy of your B cells and I eradicate your B cells, I will eradicate the malignancy as well. That doesn't work so well with lung cancer. Okay? So if you want to actually address solid tumors, tumors that people recognize with organs that everybody has and understands, then you need to find targets that are selective for tumors. They're only on tumors, and you'll see that our targets are only on the tumors that we are targeting. The other advantage is that because we use the natural targeting domain, the natural signaling machinery, we don't need to add any co-stimulatory domains. We just engineer the T-cell receptor to recognize something that it's forgotten or never knew how to recognize. And because of that, we can target a wide range of proteins because although cell surface proteins represent something like 20-something% of the overall proteome of a cell, all the proteins in a cell, those produced aberrantly by the cancer and those produced naturally, are presented for surveillance by the immune system through this mechanism that T cells recognize on this HLA peptide complex. Our lead program, as I said, targets NY-ESO. We've recently transitioned that to our partner, GSK. That happened earlier on this year. But we learned an awful lot from that program. And touching on what the learnings that we've now applied to our wholly owned pipeline, we dosed almost 100 patients across 6 indications. 4 of them were in trials that were ongoing when we handed over to GSK. We showed efficacy in 2 distinct solid tumors. For those of you who like your efficacy in scan form, this shows the impact of our T cells over a 6-month period and their ability to eradicate significant tumor masses, solid tumor masses. For those who like it in more data-y form, here's a set of spider plots. It's worth pointing out that spider plots in a metastatic synovial sarcoma setting have really not been particularly relevant prior to the introduction of our SPEAR T cells because synovial sarcoma doesn't do that. It doesn't go down. It's pretty irresponsive to most therapies. We were able to show with this that we were able to get effects across a range of preconditioning regimens with a range of target antigen expression. And really, these are very active cells. And we had breakthrough designation from the FDA. We had PRIME designation in Europe. And GSK is planning how to register this product in those jurisdictions. We also were able to show that our T cells are the agent that is doing the business. Now, in case that wasn't obvious from the previous slides, the T cells migrate to the tumor. The T cells infiltrate the tumor. The T cells inflame the tumor, even historically very cold tumors like synovial sarcoma. And they recruit other inflammatory cells. They recruit other T cells that do not target the target that we were focused on. And they also recruit macrophages. We've shown responses in 2 distinct sarcomas, and that includes patients with very low levels of NY-ESO expression, again, pointing to this effect of co-opting the rest of the immune system that our agents have. And we've been able to reduce very large tumor burdens in a way that's not been seen before. There's only one thing that we've found at this point in time that really correlates with response. It's whether you get a robust in vivo expansion. So we put the cells in, and if we see them grow, they see tumor, they grow, they multiply, they become very, very large percentage of your T cell population. And if we do that, then we see persistence, we see anti-tumor effects over months. You'll notice from the spider plot that these effects carried on over a period of 6 to 9 months in some cases. So if we see that expansion, then we are much more confident of seeing responses. And cell dose matters for that expansion. There's some form of threshold effect. You need to get to about 1 billion cells. That's a lot higher than for CAR T, primarily probably because our cells need to go and find the tumor. With a CAR T in a hematological setting, the tumor's there immediately, and the target is present in huge quantities. For us, our T cells need to do what T cells do. They go in, they work their way around the body, they find the tumor, and then they attack it. So we need more cells. And we also need appropriate preconditioning. That's true for all of the engineered T cell therapies. And that's what enables the cells, which you can really think of as a cell transplant, to engraft and to give us this expansion and then persistence. And overall, as I'll show a little bit later with some safety data, we show a promising risk-benefit profile. That's the initial program that we have. Much more exciting for us is our proprietary program coming behind, targeting 3 different targets expressed on a broad range of tumor types. MAGE-A10 on 4 tumor types. MAGE-A4 is expressed on those 9 tumors in significant percentages. And then alpha-fetoprotein, which is expressed on hepatocellular carcinoma. All of these are in phase 1-2 studies, and those of you who know anything about the engineered T-cell space know that that's when you get a signal. And we are currently walking through the dose escalation of those studies. First dose, cohort 1, was 100 million cells. That was in order to show that we didn't have some rank off target toxicity. We cleared that earlier on in the first quarter of this year. Cohort 2 was at 1 billion cells. 1 billion cells was where we started to see anti-tumor effect with our NY-ESO program. And we cleared that from a safety perspective in July and August this year for MAGE-A10 and MAGE-A4 respectively. And we're now dosing in cohort 3, which is target of 5 billion cells with a range around that representing our manufacturing capabilities. And that dosing is in progress, and we anticipate having the first scans to be able to share with the investor and scientific community later on this year. From a safety perspective, the profile that we're seeing is potentially because of the need for the T cells to go and find the tumor, significantly different to that profile that's been seen with CAR-Ts. The level of CRS is lower. The level of high-grade CRS is in the single-digit percentages, as you can see. And most of the adverse events are consistent with either the preconditioning or similar to those typically experienced by patients who have immunotherapy. Tolerability has actually been really quite good. From a patient perspective, we had a patient come and visit us, and from a patient perspective, although we don't like giving them chemotherapy as a preconditioning regimen, it's nothing compared to the chemotherapy regimens that they've taken previously. This is a really critical year for us. We've executed on everything that we said we were going to do in the first half of this year, and now the proof of the pudding, as they say, is in the eating, and that little red circle shows that we're on track for response data in our MAGE-A10, MAGE-A4 studies, and safety data from our AFP studies later on this year. And I think that'll be the proof that engineered T cells can work in solid tumors beyond what we've already shown in synovial sarcoma. So what are we doing behind that? We've set out to build a fully integrated cell therapy company, and we've made a lot of progress towards that. Initially, focused on the left-hand side of this slide, you'll see our efforts and investment in the front end, in the discovery part of this, with the TCR engineering and target selection and validation, and the preclinical testing to ensure safety prior to going into the clinic. On the right-hand side, you see the investments in the clinical testing and regulatory, where we spent most effort over the last year, consistent with a lot of comments at this conference, has been in the middle, in the manufacturing piece. The criticality of that to a capability to develop and deliver cell therapies cannot be overestimated. And so both on the vector side and on the T-cell manufacturing, we have in-house and external solutions for that will ensure that we're able to manufacture and dose the patients in our clinical trials and actually into small commercial operations initially, at least. On the right-hand side, you see our commercial. It says in progress. It's very much in progress. Kimberly, our VP of commercial, is sitting in the front row here. And the challenges in the cell therapy space are not to be sniffed at, another topic of a lot of discussion at this conference. We do that with a network of partners on the clinical side. I want to point out our relationship with MD Anderson. From a platform perspective, Universal Cells and Bellicum. And these are just a selection of our manufacturing partners, the major ones providing both vector and cell capabilities. And also, of course, our network of clinical collaborators, the leading lights of the US, Canadian, and now European oncology treatment centers. We have funding out to 2020. 129 million at the end of the last quarter. We haven't announced our financials for this year, but we did a financing a few weeks ago and raised 100 million, and we've also received milestones from GSK, so we have well north of 200 million at this point. And we have upcoming posters at both ESMO and SITC that will start to talk about the data that I referred to on the pipeline slides. So that's Adaptimmune. Leaders in TCR T-cell therapy, 3 wholly owned programs in the clinic, data later on this year. Thank you.